Thanks to the organizers for inviting us to present our data on our novel Borrelia vaccine, which we have developed over the last few years and are getting ready for clinical trials. Since I believe in the program, this is the only talk on Borrelia. I'm going to give you a few slides of introduction to the disease and to the spirochete, the bacterium causing the disease. For those of you who are not familiar with Borrelia, Borrelia is a gram-negative-like bacterium which has no LPS on the surface, but it has a lot of lipoproteins on the surface, such as OspA, which is the main topic of my talk today. There are a lot of different species around, some of which cause human disease, and I'll come back to that as well. The species of Borrelia are basically characterized by a surface molecule called the outer surface protein A. So there are several OspA serotypes, and I'll also come back to this. And importantly, the disease is transmitted by ticks, which look like this from the egg to the adult. And we'll come back to the cycle of the cycle of Borrelia within the ticks in a minute. You can see here, this is an adult female tick which can lay approximately 1,000 eggs. The eggs are normally not infected by Borrelia, so they basically have to pick up the infectious target on the first feed on small rodents or birds. From the egg, they go obviously to these animals to pick up Borrelia if these animals are infected, and only when they mold to nymphs and later on to adults, they feed on humans and can then transmit the Borrelia they picked up from these small animals also to humans. When you look at the epidemiology, there is a particular situation when you observe Lyme disease. As I mentioned initially, there are several species of Borrelia, one called Borrelia burgdorferi, second one afzelii, garinii, and lately there has been the recognition of a fourth species, bavariensis. All of them cause human disease. But when you look at the United States, you will only find Borrelia burgdorferi as a disease-causing species. There are no ticks carrying any of the other species in the United States, whereas in Europe and Asia, you find basically all four species in ticks found in nature. So all of these can cause disease with different frequencies depending on the respective countries. So if you want to develop a vaccine against Borrelia, you, depending on which region you want to go to, if you want to go to the US, you're dealing with one species, but if you want to have a broad protective vaccine that works in the US as well as in Europe, you obviously need to address all different species with all different serotypes. So obviously a multivalent approach is needed. When you look at the disease-causing species, and here is data from two strain collections, one from our competitor Baxter, one from a collaborator in Munich. You can see that these different species separated in different OspA serotypes are found to a certain frequency in human isolates and can cause invasive disease. The main species in Europe, and this is the reason why we started our work on this species, Borrelia afzelii, is of OspA serotype 2. If you want to develop a vaccine, and this is the approach that's taken also by us, and you basically include the 6 most dominant serotypes of these 4 Borrelia species, you cover in the range of at least 95% of all naturally occurring Borrelia. As I said, in the US, there is only serotype 1. These are pictures from 2001 and 2011 of only those spots, basically the regions here on the East Coast where the disease was confirmed. And just recently, approximately half a year ago, the CDC basically published preliminary numbers that the incidence of disease in the US is not as what was reported up to now in the range of 20 to 30,000 cases per year, but rather 10 times higher in the range of 300,000 cases per year, which makes it the third most reportable disease in the US after chlamydia and gonorrhea. The disease we want to prevent is not the primary infection. When you get infected by Borrelia, you often see in many cases an erythema, which looks like a bullseye, which has mild symptoms like fever, but disappears, especially if you treat it with antibiotics. If you don't notice that you are infected and you don't treat Borrelia, you can get disseminated infection to the joints, to the central nervous system, or in the skin. And these type of disease are very difficult to treat with long-term antibiotic treatment, and are the reason why there is a large need to develop a vaccine. Now, before I come to our vaccine, one more slide on the mechanism of action, which is important to understand if you want to develop an OspA vaccine. It's a very peculiar situation. OspA is expressed in the spirochetes when it's in the tick, but only when it is in the midgut of the tick, when the tick is not feeding. Once the tick starts feeding and sucks in blood, the environment changes and OspA is downregulated, which means that when Borrelia enters the human host, OspA is no longer expressed on the surface, and you cannot attack the Borrelia by an antibody or by whatever immune response that recognizes OspA. So when you develop an OspA-based vaccine, basically what you do is you immunize humans. The tick takes up the human blood, and Borrelia is basically eliminated in the tick. So the vaccine doesn't work in the human body, but it works in the tick. And actually, when we do our animal studies, we can take the ticks after the experiment and check them, and they are basically sterile. They don't have any Borrelia left anymore after treatment. So what did we do? Coming to our vaccine, we basically looked what is known about OspA, and it was reported previously that the full-length OspA molecule here shown as a crystal structure, with the N-terminal attached to the membrane, you have the main epitopes that are recognized by antibodies on the C-terminus of OspA. So we reasoned if we only take the C-terminus as our vaccine candidate, we have the relevant regions of the molecule in order to generate a properly focused immune response. We started our work with, as I said initially, just focusing on one serotype, and basically generated this region as a single protein. Now, it was also known from the literature that this molecule as such is not very stable. You cannot really use the protein. If you immunize, you get very little protection because it basically disintegrates. So what we initially did is we designed, our bioinformatician designed certain regions within the protein and said if we incorporate disulfide bridges in these regions, this would stabilize the protein and we can use it as an immunogen in mice. This was done in 5 different positions, and finally we looked at stability, we looked at initial immune responses towards this protein, and we have chosen one of these proteins which showed very good stability in vitro and also in animals. Since we wanted to generate a multivalent vaccine, we have now used the C-terminal fraction, and in order to reduce the complexity of the vaccine, since we needed 6 different serotypes, we have always taken the C-terminus of one serotype and fused it via linker to a second serotype, and on one of at the end terminus included the lipidation sequence in order to increase the immunogenicity, which then finally leads to a vaccine that is composed of 3 recombinant proteins, each of them covering 2 serotypes of Borrelia. We obviously needed to assess, is lipidation really required? We heard about lipidation yesterday. We looked at it in animal models using the monomer. Here you just see an experiment where we immunized the monomer without lipid and only half of the dose with lipid, and you see a drastic increase in immune response in mice. And this is shown on the second panel here, that this is not due to the lipid. This is the monomer without the lipid, and you see the same increase in immune response directed against the protein. So by lipidating your protein, you get a large increase, at least 10-fold increase in immunogenicity of your protein. From previous vaccine studies using OspA, it was also known that alum adjuvantation is required to increase immune response. We also did experiments using alum plus lipidation. We basically have taken here all our 3 heterodimeric proteins, always consisting of the 2 C-terminal fragments, and immunized mice at different doses, decreasing doses for all 3 heterodimers. You can see a clear response. The adjuvanted vaccine is always much more immunogenic in terms of antibody response than the non-adjuvanted version. So this gave us confidence to move ahead, saying we take those 3 proteins as a mixture, lipidated. Each of the molecules is lipidated, and we mix this with alum as the final vaccine composition. In order to compare it to what has been done previously, some of you may know that there was a vaccine in the US on the market called LYMErix. It consisted of the full-length OspA. And just for illustration purposes, I have colored this differently. The dark blue part is the part that we use in our vaccine. Down here you see our vaccine, 1 serotype, linker, and the second serotype. And there is a competitive vaccine developed by Baxter, which is basically shown here, but they use a slightly different approach. They take a full-length OspA molecule, which consists of the N-terminus fused from 1 serotype fused to the C-terminus, a small fraction of the C-terminus of the second serotype. So they have 3 molecules which are chimeric proteins and which have always the full-length protein. And we believe by including the relevant portion, the C-terminal part, which can actually be recognized by antibodies on the surface, we have the intact sequence of each serotype in the vaccine. We believe we have a distinct advantage. When you look at this in mice, and this is the data we currently have, and here we immunized mice either with the full-length OspA, the competitive product from Baxter, as good as we know, that's what's public known, and our combination. This is the full combination of the protein. You can see when you coat with the C-terminal fragment that we get a very focused immune response, which is for most of the 6 serotypes better or at least as good as the full length or the competitive product. So obviously with our vaccine at the same dose we get a better immunogenicity in terms of IgG titer. The final proof of, obviously, before we can move into the clinic, is we have to show that the vaccine is efficacious in the relevant animal model. Borrelia, there has been basically 2 models being used in the literature. One is a tick challenge model, and one is a needle challenge model. The tick challenge model had been published only for 1 serotype, so we are currently still in the process to establish further models, but we have established 2 different tick challenge models in-house and 3 different needle challenge models in-house in order to show that the vaccine works. The readout in both models is based on an ELISA against abundant protein on the surface, as well as a qPCR in order to show that mice are infected or are not infected. And just to illustrate what this implicates is when you do these experiments, you basically have to put ticks on the back of the mice and let them feed almost for a week. And after a week, you have to observe whether the ticks have fully fed, and only those mice where at least one tick has fully fed are included in the experiment. So if this container falls off, or if no tick has fed, obviously the mouse will not be included in the experiment. That's important, and that's what you also see in the protection data. For the first model we have established is the serotype 2, the Borrelia afzelii, and what you can see here is we have immunized mice, either with a full-length OspA as a control, 1 microgram, fairly low dose, with a competition 3 microgram, meaning 1 microgram of each of the 3 proteins, or our combination 3 microgram, again each 1 microgram of the 3 proteins. And as you can see, 3 different independent experiments, and each group normally contained 10 mice, but as we only count those which have a fully fed tick, the numbers deviate from 10 because some of the mice dropped out. But in all of these experiments, we see very nice protection. All of the mice with each of the 3 vaccines were fully protected, whereas the placebo, no vaccine, they were almost all infected. We have established the same type of data against the serotype 1. This is the main species that is circulating in the US. And again, you can see here that with a similar dose, you get full protection with the 3 vaccine, making us very confident that the vaccine will also deliver in humans. Currently, we have a very comprehensive data set in terms of protection. We have basically for serotype 1, as I just shown you, the tick challenge model, as well as a needle challenge model, and we always have at least 3 experiments, independent experiments, showing significant levels of protection. We have a tick challenge model, as I've shown you, for serotype 1, and we have 2 needle challenge models for serotype 5 and 6, showing that you have full protection with our vaccine. In terms of the remaining 2 serotypes, which is serotype 3, shown here, and serotype 4, shown here, we are still working on the animal model. It's fairly difficult to get Borrelia species into ticks and get them infectious, because mice are normally not readily infected by Borrelia. But there is some first data that this seems to be possible. In the meantime, we basically look for surface staining of the induced antibodies in live Borrelia, and this is what you can see here. When we compare this to the competitive products, we see a very similar surface staining, or we do a growth inhibition assay as shown here for serotype 4, where you can see that we are very competitive compared to the other products. And in terms of protection, you see basically here a summary for the other 4 serotypes. We have very good protection for all 4 other serotypes. So we are basically now in the position to, and this is only for illustrative purposes, to move into a phase 1 study. We are almost there, and we are currently discussing how we're going to move this ahead in order to test this clinically, because obviously there is a need to develop a vaccine potentially differently for the US and for Europe, because of the existence of different serotypes circulating in these 2 territories. So in summary, what I've shown you is the protein-based vaccine. What we have developed can protect against all different serotypes that are currently circulating or the major 6 that are currently circulating in Europe and in the US. We have set up with an enormous amount of work, 5 different animal models showing that this works. What I haven't shown you is that we can produce the protein in excellent quality, yielding, at the moment, we are in the range of roughly 1 gram of mature lipoprotein per liter of fermentation. The 3 proteins can be produced with a very similar process. I call it here semi-generic process, and it's scalable, so we can basically readily transfer this process to a GMP facility, and we anticipate that the project should be ready to enter phase 1 within approximately a year. And with this, I'm basically at the end. We did a lot of challenge studies, but the people in the lab also like to be challenged themselves, as you can see on the picture. This is the preclinical research group from Vienna who did most of the work. The group leader from Sweden, actually 2 people. One is not on the picture here, Per Komstedt, who together with Urban Lundberg led most of the studies. And we had a number of collaborators externally, Sven Bergström from Sweden, Hans Daudl, with whom we basically set up the tick challenge model, because he is capable of culturing ticks in the laboratory and infecting them with distinct species of Borrelia, and Volker Fingerle in Munich, who supplied some of the strains. And with this, I'm happy to take any questions if you have.
Thanks, Andreas. So we have just time, Ursula.
I'm just wondering, I'm not so familiar with that, but as I remember, the American Lyme disease vaccine was taken off from the market due to side effects leading to arthritis. So I don't know exactly the mechanism that was causing this, and is this still an issue in your composition of the vaccine?
Well, to the current knowledge, the vaccine was not taken off because of the side effects, but obviously there was speculation that there was a human epitope, the LFA1 epitope, that potentially caused this. This was never proven, and it was never. Was actually the researcher who discovered it later on took it basically back. But to be on the safe side, Baxter's as well as our vaccine has eliminated this epitope, and this is only present in one of the OspA serotypes, namely serotype 1, and we have mutated this single epitope in order to avoid any potential cross-reactivity that it could elicit in humans.
Thanks again, Andreas, because we are running a little bit late, and so this was a nice example of vaccine design, and thanks for sharing with us.